So let's walk through a primer on neuroembryology, embryology of the nervous system. These are the key tutorials that we'll cover. So we'll start with neurulation. We're starting from this trilaminar germ disc, which comprises, remember, ectoderm, mesoderm, and endoderm. These are the key steps. The notochord from the mesoderm is going to induce the overlying ectoderm to form a neural plate. Then the neural plate is going to fold into a neural tube, and neural crest cells are going to pinch off. The tube is going to become the central nervous system, and the crest cells are going to form the peripheral nervous system, and then some select other structures. And we're going to hammer this again and again throughout this session. So here's our trilaminar germ disc in coronal view, and then this is an axial view of the ectoderm here. So again, notochord within the mesoderm induces this ectoderm to form this neural plate here. Neural plate folds into this neural tube. We can see this folding pattern here. Neural crest cells dorsally, and then these somites flanking the neural tube. And then on day 24, this anterior cranial neuropore closes, and day 26, this posterior or caudal neuropore closes. The neural tube itself, its internal organization. So this is the end product. This is the spinal cord here. So the neural plate has an alar plate dorsally, forms sensory, basal plate anteriorly, forms this ventral horn, motor function. So again, notochord within mesoderm induces overlying ectoderm to form the neural plate. Plate starts to fold, folding becomes this neural tube, and then these neural crest cells pinch off, and we'll talk about those in detail in a little bit. Here is a blown up view here of this neural tube organization. Again, alar plate, dorsal horn, sensory, basal plate, ventral horn, motor. And then we talked about these somites. So the somites differentiate into sclerotome, think bone, myotome, think muscle, and then dermatome, think dermis. Neural crest cell differentiation. These are some core divisions of, or core differentiating structures from the neural crest cells. There's a peripheral nervous system, which we've mentioned, the meninges, melanocytes, so pigmentary cells of the skin, and then the enteric nervous system, the gastrointestinal system. So a wide variety of structures, and that's what makes it challenging to remember all this. We can think about this in terms of different anatomical divisions. There's the cranial, which is these pharyngeal arch components, which we'll talk about again in a minute. And then at the trunk and lumbosacral divisions, there's the dorsal root ganglia, sympathetic chain, adrenal medullary cells, and prevertebral ganglia. So this is an illustration to help show the brain vesicles, which we'll talk about momentarily, and then the neural crest cells in mid-view here in midline dorsally, and then the somites flanking the neural tube. Again, notochord induces ectoderm to form neural plate. There's folding that forms the neural tube. Here's the neural crest cells. They pinch off and make their great migration, and then the somites flank the neural tube here. So let's talk about this great migration that these neural crest cells make. So we said pharyngeal arch components, so they form musculoskeletal structures and the cranial nerves of the peripheral nervous system here. Then they also, during this migration, form this dorsal root ganglia at the trunk and lumbosacral and cervical regions. So we can see this dorsal root ganglion here. It houses the sensory cell body. Then it migrates and forms the sympathetic chain here. This is the autonomic nervous system, sympathetic division. Again, remember parasympathetic, rest and digest, sympathetic for activation, fight or flight. Adrenal medullary cells, these chromaffin cells, it also forms, and then it forms this prevertebral ganglia here. And then it forms the enteric nervous system. So it also forms the nervous system to the digestive tract. And then don't forget the meninges and melanocytes. So a lot of different structures coming from the neural crest cells. Brain vesicle formation, so patterning and organization of the brain. At 3 weeks, there's 3 vesicles or cephalon. There's the prosencephalon, mesencephalon, and rhombencephalon. And then at 5 weeks, there's 5 of them. So there's a telencephalon, diencephalon, mesencephalon remains, and then the rhombencephalon forms the metencephalon and the myelencephalon. We can see this here. These are the primary brain vesicles at 3 weeks. And then we can see this division of the prosencephalon to the telencephalon and diencephalon, and the rhombencephalon divides to the metencephalon and myelencephalon, and then there's the neural tube. So what do these things become? So the telencephalon becomes the cerebrum, the diencephalon becomes the thalamic areas, the mesencephalon forms the midbrain, and then the metencephalon forms the pons and cerebellum, and then the myelencephalon forms the medulla, and then the neural tube, the spinal cord. And we can see this within our fetus here, differentiated. This is this cephalic flexure. When we talk about orientational planes of the cerebrum, there is an angulation that the brainstem takes to it, and that's why these terms rostral, caudal, dorsal, ventral differ from the brain to the brainstem. Finally, the optic vesicle forms an optic cup, which forms the retina here, and then has an optic stalk, which forms the optic nerve here. So that's some key embryology. That's where the nervous system comes from. And when you study this in detail, you can really get a sense of how things pattern and how they organize themselves.